Welcome back, everyone. We continue our discussion on management of diabetes in pregnancy, clinical update. Today's topic is pathophysiology of gestational diabetes mellitus. Now, what is GDM? GDM is a form of hypoglycemia. Similar to other forms of hyperglycemia, GDM is a disease of pancreatic beta cells which do not produce sufficient insulin to meet the increased requirements of late pregnancy. The simplicity of this description belies a more complex set of etiologies for GDM. Mechanistic studies of GDM reveals at least 3 separate underlying causes of beta cell dysfunction. First, some women have circulating immune markers, for example, anti-islet cell antibodies or antibodies to glutamate decarboxylase 65 that are diagnostic of evolving type 1 diabetes mellitus. The frequency is generally less than 10% of all women with GDM, and it tends to parallel the background prevalence of type 1 diabetes mellitus in the population. Second, some women have genetic variants that are diagnostic of monogenic forms of diabetes. This includes genes for subtypes of maturity onset diabetes of the young, or MODY, and maternally inherited diabetes. Systematic data on the frequency of these monogenic forms of diabetes in GDM are limited, but they appear to be infrequent, accounting for 1 to 5% of cases. The third general setting in which the beta cell defects that underlie GDM occur is that of obesity and chronic insulin resistance. This group represents a majority of cases of GDM, leading many clinicians to view GDM as a form of evolving type 2 diabetes mellitus. This view may be accurate for the majority of cases, but the full spectrum of GDM includes other causes of inadequate beta cell function in relatively young women. Appropriate care for mothers, especially after pregnancy, requires an understanding of this fact, as will be discussed later. Understanding of the role of acquired insulin resistance of pregnancy in the pathogenesis of the hyperglycemia that defines GDM has evolved over time. Traditionally, it's been taught that GDM develops when beta cells fail to keep pace with the increasing insulin resistance that occurs during the second half of pregnancy. The resultant increasing imbalance between insulin demand and supply manifests itself as rising glucose levels, especially during the second half of pregnancy when insulin resistance is the greatest. In this scenario, glucose regulation returns to normal postpartum only to resurface years later as impaired glucose levels and diabetes mellitus, usually type 2 diabetes mellitus. Serial studies of insulin resistance and beta cell function in women who develop GDM provide quite a different picture. A large majority of the insulin secretory defect that is present in the third trimester of pregnancy is present before and soon after pregnancy. In fact, insulin secretion during pregnancy increases in parallel in women with and without GDM, but from a lower starting point in women with the condition. Clinical characteristics of the women in these studies suggest that they fall into the subtype of GDM that's related to type 2 diabetes mellitus. Thus, many and perhaps most women with GDM appear to have a beta cell defect that is chronic rather than acquired during pregnancy. This concept is consistent with the fact that GDM tends to occur in women over 30 years old who have had multiple pregnancies and who are obese. Obesity and pregnancy are conditions that promote chronic beta cell dysfunction that is detected during pregnancy when glucose tolerance is tested for the first time in many women's lives. In summary, GDM is a form of generally mild hyperglycemia that reflects inadequate beta cell compensation for the body's insulin needs. In some cases, the acquired insulin resistance of pregnancy could create insulin demands that exceeds beta cell capacity to supply insulin for the limited time frame of pregnancy. However, most cases appear to represent chronic beta cell dysfunction that is detected during pregnancy when glucose tolerance is measured as part of routine care and often for the first time in a young woman's life. As discussed later, beta cell function is not just deficient during pregnancy in women with GDM. It deteriorates over time, which results in women who have had GDM being at high risk of developing diabetes mellitus in the years following the index pregnancy. Now, GDM is due to inadequate insulin response to normal pregnancy-induced insulin resistance. In normal pregnancy, maternal tissues become progressively insensitive to insulin. This is believed to be caused partly by hormones from the placenta and partly by other obesity and pregnancy-related factors that are not fully understood. Skeletal muscle and adipose tissue are the main whole-body glucose disposable sites. In normal pregnancy, insulin-mediated whole-body glucose disposal decreases by 50%. In order to maintain an euglycemic state, the woman must increase her insulin secretion by 200 to 250%. GDM develops when the pregnant woman is not able to produce an adequate insulin response to compensate for the normal insulin resistance. GDM is observed in obese as well as in lean women. However, the pathophysiology behind the disease is believed to differ between these groups. In obese women, the pathophysiology is primarily characterized by the pregnancy-induced insulin resistance being amplified by the already elevated pre-pregnant insulin resistance level. The elevated insulin resistance level is a known factor in the metabolic syndrome. In lean women, the same factors appear to play a role, but the defect in the first phase insulin response contributes to a larger extent. These defects culminate in a disruption of the action of insulin in maintaining glucose levels, resulting in maternal hyperglycemia. Glucose is transferred via the placenta to the fetus. Maternal hyperglycemia therefore stimulates a fetal hyperinsulinemia to counter this excess placental glucose transfer. The high insulin level in the fetus stimulates growth, which results in fetal macrosomia, or birth weight over 4,000 grams. Let's now discuss glucose regulation during healthy pregnancy. During healthy pregnancy, the mother's body undergoes a series of physiological changes in order to support the demands of the growing fetus. This includes adaptations to the cardiovascular, renal, hematologic, respiratory, and metabolic systems. One important metabolic adaptation is in insulin sensitivity. Over the course of gestation, insulin sensitivity shifts depending on the requirements of pregnancy. During early gestation, insulin sensitivity increases, promoting the uptake of glucose into adipose stores in preparation for the energy demands of later pregnancy. However, as pregnancy progresses, the surge of local and placental hormones, including estrogen, progesterone, leptin, cortisol, placental lactogen, and placental growth hormone, together promotes a state of insulin resistance. As a result, blood glucose is slightly elevated, and this glucose is readily transported across the placenta to fuel the growth of the fetus. The mild state of insulin resistance also promotes endogenous glucose production and the breakdown of fat stores, resulting in a further increase in blood glucose and free fatty acid concentrations. Evidence in animals suggests that in order to maintain a glucose homeostasis, pregnant women compensates for these changes through hypertrophy and hyperplasia of pancreatic beta cells, as well as increased glucose-stimulated insulin secretion. The importance of placental hormones in this process is exemplified by the fact that maternal insulin sensitivity returns to pre-pregnancy level within a few days of delivery. For reasons that will be explored in this update, the normal metabolic adaptations to pregnancy do not adequately occur in all pregnancies, resulting in GDM. We will now go into detail on the pathophysiology of gestational diabetes mellitus. We'll be discussing molecular processes underlying the pathophysiology of GDM. GDM is usually the result of beta cell dysfunction on the background of chronic insulin resistance during pregnancy, and thus, both B cell impairment and tissue insulin resistance represent critical components of the pathophysiology of GDM. In most cases, these impairments exist prior to pregnancy and can be progressive, representing an increased risk of type 2 diabetes mellitus post-pregnancy. The number of additional organs and systems contribute to or are affected by GDM. This includes the brain, adipose tissue, liver, muscles, and placenta. First is beta cell dysfunction. The primary function of beta cells is to store and secrete insulin in response to glucose load. When beta cells lose the ability to adequately sense blood glucose concentration or to release sufficient insulin in response, this is classified as beta cell dysfunction. Beta cell dysfunction is thought to be the result of prolonged excessive insulin production in response to chronic fuel excess. However, the exact mechanisms underlying beta cell dysfunction can be varied and complex. Defects can occur at any stage of the process: pro-insulin synthesis, post-translational modifications, granule storage, sensing of blood glucose concentrations, or the complex machinery underlying exocytosis of granules. Indeed, the majority of susceptibility genes that are associated with GDM are related to beta cell function, including the potassium voltage-gated channel KQT-like 1 or KCNQ1 and glucokinase or GCK. Minor deficiencies in the beta cell machinery may also be exposed in times of metabolic stress such as pregnancy. Beta cell dysfunction is exacerbated by insulin resistance. Reduced insulin-stimulated glucose uptake further contributes to hyperglycemia, overburdening the beta cells, which have to produce additional insulin in response. The direct contribution of glucose to beta cell failure is described as glucotoxicity. Thus, once beta cell dysfunction begins, a vicious cycle of hyperglycemia, insulin resistance, and further beta cell dysfunction is set in motion. Animal studies suggest that beta cell number is also an important determinant of glucose homeostasis. First, the short-term but dramatic reduction in beta cell mass overburdens the remaining beta cells, resulting in severely reduced glucose-stimulated insulin secretion and the depletion of internal insulin granule stores. Second, the loss of beta cell mass has been linked to epigenetic downregulation of the pancreatic homeobox transcriptor factor PDX1, which is essential for normal beta cell differentiation in the embryo. Prolactin is also essential for adequate beta cell proliferation as demonstrated in mouse knockouts of the prolactin receptor. In addition, glucotoxicity is also thought to result in beta cell apoptosis over time. Pancreatic samples of type 2 diabetes mellitus patients can show a reduction of beta cell mass by 40 to 60%, but less than 24% loss after 5 years of disease has also been reported. Reduced beta cell hyperplasia may also play a role in GDM, based on animal studies and limited post-mortem human studies. Therefore, reduced beta cell mass, reduced beta cell number, beta cell dysfunction, or a mix of all three contribute to GDM, depending on the individual. Next is chronic insulin resistance. Insulin resistance occurs when cells no longer adequately respond to insulin. At the molecular level, insulin resistance is usually a failure of insulin signaling, resulting in inadequate plasma membrane translocation of glucose transporter 4 or GLUT4, the primary transporter that is responsible for bringing glucose into the cell to use as energy. The rate of insulin-stimulated glucose uptake is reduced by 54% in GDM when compared with normal pregnancy. While insulin receptor abundance is usually unaffected, reduced tyrosine or increased serine-threonine phosphorylation of the insulin receptor dampens insulin signaling. In addition, altered expression and/or phosphorylation of downstream regulation of insulin signaling, including insulin receptor substrate or IRS1, phosphatidyltree kinase or PI3K, and GLUT4 has been described in GDM. Many of these molecular changes persist beyond pregnancy. Several of the previously discussed risk factors for GDM are thought to exert their effects by interfering with insulin signaling. For example, saturated fatty acids increase intracellular concentration of diacylglycerol with myocytes, activating protein kinase C, PKC, and inhibiting tyrosine kinase, IRS1, and PI3K. Pro-inflammatory cytokines and Adiponectin also modify this process, as discussed later. Third is neurohormonal networks. Neurohormonal dysfunction has been implicated in the pathogenesis of diseases of insulin resistance, such as that present in GDM. This network regulates appetite, active energy expenditure, and basal metabolic rate, and it is made up of a complex network of central, for example, cortical centers that control cognitive, visual, and reward cues, and peripheral, for example, and satiety and hunger hormone signals. These contribute to GDM by influencing adiposity and glucose utilization. The network is highly regulated by the circadian clock, which may explain why pathological sleep disorders or those individuals undertaking shift work are correlated with GDM rates. Neural networks controlling body weight are most likely set in early life, as demonstrated in animal studies. For example, rats that are both under and overfed in early life experience epigenetic alteration of the regulatory set point of hypothalamic neurons. This adds to the previously mentioned suggestion that predisposition to GDM may be set in the womb. Some of the most important regulators of neurohormonal metabolic control are adipokines, cell-signaling proteins that are secreted primarily by adipose tissue. These include leptin and adiponectin. Let's talk about leptin. Leptin is a satiety hormone secreted primarily by adipocytes in response to adequate fuel stores. It primarily acts on neurons within the arcuate nucleus of the hypothalamus to decrease appetite and increase energy expenditures. Specifically, leptin inhibits appetite stimulators, neuropeptide Y or NPY, and agouti-related peptide or AGRP, and it activates the anorexigenic polypeptide pro-opiomelanocortin or POMC. When leptin was first discovered, it was lauded as a potential treatment for obesity. However, it was soon revealed that the majority of obese individuals do not respond to leptin and instead demonstrate leptin resistance. While leptin treatment is effective in obesity that is caused by leptin and leptin receptor genetic polymorphisms, these are rare, less than 5% of obese individuals. Therefore, obesity is associated with excessive plasma leptin concentration or hyperleptinemia as a result of leptin resistance, and plasma leptin concentrations are generally proportional to the degree of adiposity. Leptin resistance can occur either as a defect in blood-brain barrier leptin transport or through intracellular mechanisms that are similar to insulin resistance. Like insulin resistance, a degree of leptin resistance occurs in normal pregnancy, presumably to bolster fat stores beyond what would usually be required in the non-pregnant state. Leptin resistance is further increased in GDM, resulting in hyperleptinemia. However, pre-pregnancy body mass index is a stronger predictor of circulating leptin than GDM per se. The placenta also secretes leptin during human pregnancy. In fact, the placenta is responsible for the majority of plasma leptin during pregnancy. Placental leptin production is increased in GDM, probably as a result of placental insulin resistance, and this further contributes to hyperleptinemia. This is also thought to facilitate amino acid transport across the placenta, contributing to fetal macrosomia. Next is adiponectin. Similar to leptin, adiponectin is a hormone that is primarily secreted by adipocytes. However, plasma adiponectin concentrations are inversely proportional to adipose tissue mass, with low concentrations in obese individuals. GDM is similarly associated with decreased adiponectin. In contrast to leptin, there is a stronger association of adiponectin with insulin resistance than with adiposity. This suggests that adiponectin plays an important role in the pathogenesis of GDM independent of obesity. Now, adiponectin enhances insulin signaling and fatty acid oxidation, and it inhibits gluconeogenesis. It does so by activating AMP-activated protein kinase or AMPK within insulin-sensitive cells, which facilitates the action of IRS1, and by activating the transcription factor peroxisome proliferator activated receptor alpha or PPAR alpha in the liver. Furthermore, adiponectin stimulates insulin secretion by upregulating insulin gene expression and exocytosis of insulin granules from beta cells. Adiponectin is also expressed in low concentration from the syncytial trophoblast of the placenta, where it is regulated by cytokines such as tumor necrosis factor alpha or TNF-alpha, interleukin-6 or IL-6, interferon gamma, and leptin. The role of placental adiponectin in normal and GDM pregnancy is unclear. However, emerging evidence suggests adiponectin impairs insulin signaling and amino acid transport across the placenta, limiting fetal growth. Therefore, adiponectin gene methylation in the placenta is associated with maternal glucose intolerance and fetal macrosomia. Fourth is adipose tissue. Originally believed to exist only as a passive depot of energy, the discovery of leptin in 1994 established adipose tissue as an essential endocrine organ. Adipose tissue both ensured that energy is partitioned safely and it actively secretes circulatory factors, including adipokines, the aforementioned leptin and adiponectin, and cytokines such as TNF-alpha, interleukin-6, and interleukin-1 beta, which have wide-ranging metabolic effects. Adipose tissue for energy storage. The storage capability of adipose tissue is essential for metabolic health. This is exemplified through two extremes. Rare disorders in which white adipose tissue is absent lead to severe metabolic syndrome, whereas some obese individuals with excessive white adipose tissue do not develop metabolic syndrome at all. Therefore, the ability to partition excess calories into adipose tissue rather than ectopically in the liver, muscle, or pancreas appeared to serve as a protective measure. Non-diabetic obese individuals exhibit adequate adipose tissue expansion in response to fuel surfeit, and therefore maintain healthy blood glucose concentration, sufficient beta cell compensation, and avoid chronic insulin resistance. In this way, organs avoid glucose and fatty acid-induced tissue damage. As previously mentioned, early pregnancy is marked by an increase in adipose tissue mass, which later pregnancy promotes the mobilization of fats from adipose tissue in order to fuel fetal growth. Both of these processes are thought to be limited in GDM. GDM is associated with reduced adipocyte differentiation and increased adipocyte size or hypertrophy, accompanied by downregulated gene expression of insulin signaling regulators, fatty acid transporters, and key adipogenic transcription factors such as PPAR gamma. The combination of insulin resistance and reduced adipocyte differentiation hinders the tissue's ability to safely dispose of excess energy, contributing to gluco and lipotoxicity in other peripheral organs. Indeed, both type 2 diabetes mellitus and gestational diabetes mellitus are associated with lipid deposition in muscle and liver. Now let's discuss adipose tissue inflammation. Obesity, type 2 diabetes mellitus, and gestational diabetes mellitus are associated with an increased number of resident adipose tissue macrophages that secretes pro-inflammatory cytokines, including TNF-alpha, interleukin-6, and interleukin-1 beta. The importance of a low-grade inflammatory state in the pathogenesis of insulin resistance has recently become apparent. Pro-inflammatory cytokines have been discovered to both impair insulin signaling and inhibit insulin release from beta cells. These factors induce insulin resistance either by diminishing insulin receptor tyrosine kinase activity, increasing serine phosphorylation of IRS1, or through the STAT3-SOCS3 pathway, which degrades IRS1. Circulating concentrations of pro-inflammatory cytokines are increased in GDM. Plasma TNF-alpha, in particular, is strongly correlated with insulin resistance. Similarly, placental gene expression of TNF-alpha, interleukin-1 beta, and the receptor has been reported to be increased in GDM. However, the relationship between pregnancy and inflammation is complex. For example, Adipose et al. Reported that GDM placenta secrete fewer pro-inflammatory cytokines than healthy placenta. This suggests that while chronic low-grade inflammation appears to be important in the pathogenesis of GDM, the relationship may not be straightforward. Now, fifth is the liver. GDM is associated with upregulated hepatic glucose production, or gluconeogenesis. Gluconeogenesis is increased in the fasted state and not adequately suppressed at the fed state. This is not believed to be entirely the result of inaccurate glucose sensing due to insulin resistance, as the majority of glucose uptake by the liver, around 70%, is not insulin dependent. Common factors between the insulin signaling pathway and the pathways controlling gluconeogenesis, such as PI3K, might contribute to these effects. Increased protein intake and muscle breakdown may also stimulate the process by providing excess gluconeogenesis substrate. Despite this, the liver does not seem to be a primary pathogenic driver of type 2 diabetes mellitus or gestational diabetes mellitus. 6 is skeletal and cardiac muscle. Traditionally, skeletal muscle insulin resistance was believed to play a causal role in type 2 diabetes mellitus. However, skeletal muscle insulin resistance now appears to be a consequence of hyperglycemia, a protective measure to prevent metabolic stress and steatosis. Even following a short period of overfeeding, cardiac and skeletal muscle develop insulin resistance in order to divert the excess energy into adipose tissue. This is an important distinction when considering potential treatment for GDM. Attempts to directly reverse skeletal muscle insulin resistance without reducing plasma glucose concentration could be detrimental. Separate to insulin sensitivity. Type 2 diabetes mellitus and gestational diabetes mellitus are associated with a reduced number and function of mitochondria within skeletal muscle cells. This could be the result of genetics, early life programming, or chronic inactivity. Therefore, decreased number and function of mitochondria is likely an additional contributor to reduce glucose utilization in GDM. Seventh is gut microbiome. There is emerging evidence that microbial organisms within the gut, called the gut microbiome, might contribute to metabolic disease, including GDM. The gut microbiome can be influenced by early life events, such as preterm delivery and breastfeeding, and by events in later life, such as diet, composition, and antibiotic use. The gut microbiome has been consistently reported to be different between metabolically healthy and obese individuals, including during pregnancy. Furthermore, a study of stool bacteria in women with a past case of GDM reported a lower proportion of the phylum Firmicutes and higher proportion of the family Prevotelaceae as compared with normoglycemic pregnancy. Similar associations have been observed in obesity, type 2 diabetes mellitus, fatty liver disease, and elevated total plasma cholesterol. Firmicutes metabolize dietary plant polysaccharides. This may explain some of the dietary risk factors for GDM that are discussed earlier. Both red meat and animal protein decreases levels of firmicutes, while high dietary fiber increases them. However, the findings by Fugman et al. In 2015 remained after adjustments for dietary habits. Therefore, firmicutes appear to be relevant to pathogenesis of GDM independent of the diet, although the mechanisms underlying this are unknown. Prevotelaceae are mucin-degrading bacteria that may contribute to increased gut permeability. Gut permeability is regulated by tight junction proteins such as zonulin or ZO1. Increased free plasma serum ZO1 is associated with type 1 diabetes, type 2 diabetes, and GDM. Increased gut permeability is thought to facilitate the movement of inflammatory mediators from the gut into the circulation, promoting systemic insulin resistance. 8 is oxidative stress. Oxidative stress describes an imbalance between pro-oxidants and antioxidants in cells. Oxidative stress can lead to cellular damage by interfering with the state of proteins, lipids, and DNA, and has been implicated in the pathogenesis of many diseases, including GDM. Reactive oxygen species, or ROS, are described as free radicals and non-radical derivatives of oxygen and include superoxide anion, hydroxyl radical, and hydrogen peroxide. A hyperglycemic environment is associated with oxidative stress, and GDM women have been reported to overproduce free radicals and have impaired free radical scavenging mechanisms. ROS inhibits insulin-stimulated glucose uptake by interfering with both IRS1 and GLUT4. ROS also slows glycogen synthesis in the liver and muscle. Pro-inflammation cytokines, such as TNF-alpha, may also contribute to oxidative stress by increasing the expression and the activation of ROS precursors like NADPH oxidase 4 or NOX4. Interestingly, iron supplementation in women already replete in iron is associated with GDM. Several studies suggest that this relationship is the result of increased oxidative stress. Iron is a transitional metal and it can catalyze the reaction from O2 negative and HCO2 to extremely reactive OH ions within mitochondria. On the contrary, selenium and zinc are transitional metals that are necessary for the activity of some antioxidant enzymes, which may explain their inverse association with GDM. Homocysteine, a non-protein alpha amino acid that is formed by the demethylation of methionine, is also thought to contribute to GDM via oxidative stress. Exposure of beta cells to even small amounts of homocysteine results in dysfunction and impaired insulin secretion. A recent meta-analysis examined the relationship between serum homocysteine concentration and GDM in 10 eligible studies. The authors report significantly higher homocysteine concentrations among women with GDM as compared with those without GDM. B vitamins including folic acid, B2, B6, and B12 are essential for homeostasis, and this may be one reason why deficiencies and imbalance of these micronutrients are associated with GDM. And lastly, placental transport. The placenta contributes to insulin resistance during pregnancy via its secretion of hormones and cytokines. As the barrier between the maternal and fetal environments, the placenta itself is also exposed to hyperglycemia and its consequences during GDM. This can impact transport of glucose, amino acids, and lipids across the placenta. First, glucose. Glucose is the primary energy source for the fetus and the placenta, and therefore must be readily available at all times. For this reason, insulin is not required for the placental transport of glucose. Instead, glucose transport occurs via GLUT1 by carrier-mediated sodium-independent diffusion. However, the placenta still expresses the insulin receptor, and insulin signaling can influence placental metabolism of glucose. The receptiveness of the placenta to glucose uptake means that it is particularly sensitive to maternal hyperglycemia, and this directly contributes to increased fetal growth and macrosomia. Next is protein. Amino acid transport across the placenta is also an important determinant of fetal growth. GDM is associated with increased system A and L activity. This can also be modulated by pro-inflammatory cytokines such as TNF-alpha and interleukin-6. Altered amino acid transport may also be one mechanism of which excess protein intake contributes to GDM. And lastly, lipids. Finally, while GDM has traditionally been described as a disease of hyperglycemia, the rise in obesity-associated GDM has prompted a greater focus on the role of hyperlipidemia in GDM. The majority of placental gene expression alterations in GDM occur in lipid pathways, or 67%, as compared with glucose pathways, 9%. Preferential activation of placental lipid genes is also associated with GDM compared with type 1 diabetes mellitus. These data correlate with the results of the HAPO study, which revealed independent effects of maternal obesity and glucose on excessive fetal growth. Therefore, it appears that GDM influences the placental transport of glucose, amino acids, and fatty acids, and that all three must be considered when discussing the impact of GDM on placental function and fetal growth. In addition to these alterations in placental transport, GDM has been associated with other changes in the placenta. Some recent studies have reported that GDM is associated with placenta global DNA hypermethylation. Similarly, studies of the placental proteome have identified differences in the expression of proteins between GDM and non-GDM placenta. However, more research is required before the role of placental epigenetic and proteomic modifications in GDM is fully understood. There has also been recent interest in small, non-coding single-stranded segments of RNA called microRNAs, or miRNAs, expressed in placental trophoblast cells. miRNAs are involved in a number of cellular processes, including proliferation, differentiation, and apoptosis. Emerging evidence suggests that exosomes containing miRNAs are shed from the placenta during gestation and released into the maternal circulation, which can in turn influence the functioning of other cells, potentially contributing to the pathogenesis of GDM. Interestingly, exposure to endocrine disrupting chemicals, or EDCs, including bisphenol A, found in food packaging materials and consumer products, has been associated with GDM, and it has been suggested that this could be because EDCs induce exosome signaling from the placenta. Interestingly, EDCs include PPA have also been associated with alterations in methylation, perhaps linking the two mechanisms. This ends our discussion on the pathophysiology of GDM. See you next time when we discuss the risk factors in developing GDM.